last year. It's
Bailey is from Montreal. I was thinking, oh yeah, that's good. Well, never heard of him. Yeah.
The Ryan from it's the current head of one case. Fence in Mexico. So you might know.
Right, yeah. Richard said he's leaving.
So I wait till 5 plus to start.
Yeah.
So usually it's an hour to answer questions and then
It could be an hour. So, you know, well, no, it's good if you say an hour, I'll try to
yeah, we'll just end and really get time for some questions after. Yeah, we've got tea and coffee and mingle naturally. That's important. I'm just the lights at the back of so do something on it. I'm going to ask them to sit. Okay, good evening. Welcome to the School of Geography and to this, the first of this year's GA lectures, so it's great to see you all here. Thanks for coming. My name is Matthew Jones, I'm one of the members of staff in the School of Geography here, and I just get the nice job of introducing my colleague Suzanne McGowan to you today and just kicking things off. And Mary, who many of you know, will say a few words at the end of today's lecture. So it's a real pleasure to have Suzanne talking to us today. I'm really lucky in that once I managed to get on a field trip with Suzanne to this part of the world, and it is absolutely incredible, stunning place to go and work and visit. And Suzanne has been doing some absolutely outstanding, awesome work out there. So really lucky to have her talking to us today, really looking forward to it. I'll leave you in her very capable hands.
Well, thanks very much, Matt, for that. I've really got to aim high now. So I hope you find this work interesting and outstanding. And yeah, as Matt said, he was there in the early days when I'd started off working at Nottingham and came out to some of our earlier field trips. And so I've probably been working in Greedon for about 18 years now altogether. And this talk will be more generally about the Arctic and some of the recent work we've been doing. So feel free to tweet, Facebook, whatever. It's not even Instagram, is it now? I don't know. Feel free to do social media. So, okay then. So I'm going to give a little bit of background as to why we've been working on the Arctic carbon cycle. And of course, this is incredibly important for climate change generally. And generally speaking, the oceans are the Earth's greatest carbon sink, of course, which I'm sure you're all aware of. But there's a phenomenon where there's a seasonal rise and fall of carbon emissions, which correlates with this so-called summer greening in northern ecosystems. And of course, that is because during most of the winter, the Arctic's in darkness and things don't grow very much. Whereas once they start to grow, they can start to take up carbon dioxide and cause this kind of seasonal change. So the Arctic and the way that it functions is incredibly important to the carbon cycle overall, and therefore to how carbon emissions kind of get processed on a global scale. Generally speaking, the Arctic has been a sink for atmospheric carbon dioxide since the last ice age. So it's one of the world's important carbon sinks, and we need to know how it's going to respond in the future to global climate change. So let's think about why the Arctic, or the features of the Arctic that are important in terms of being the Earth's carbon sink. Well, part of that carbon is locked up in the atmosphere. Actually, this is just a little note to give you some idea of the terminology. When we talk about carbon, I'm going to use this term petagram, which you may or may not be familiar with. This is a 1/4 of a gram. Obviously we're talking about a lot of grams when we're measuring carbon on a global scale. And if we think about petagrams in the atmosphere, above 60 degrees north, we have 125 petagrams of carbon that exists in the atmosphere. That compares to 750 petagrams globally. The reason why oceans are important in the Arctic, I won't be talking about oceans for much of this talk, but generally speaking, the cold temperatures in the Arctic are very important because colder temperatures are more efficient for carbon dioxide uptake and dissolution. So they're efficient for carbon uptake, and it's thought that, or the estimates are that the Arctic Ocean is responsible for 5 to 14% of the Earth's ocean carbon uptake, but that's based on only 3% coverage of the Earth's oceans. So they punch above their weight in terms of carbon uptake. Now, what I'm going to spend most of the lecture talking about today is what goes on land in Arctic regions. It's where most of my work's been conducted, and of course lakes are part of the land kind of system. And this is probably the part of the global system that is more difficult to understand because we don't have a lot of basic data to understand how Arctic systems will respond. And one of the really sort of key research areas right now is going on in tundra regions because it's thought to be a very important carbon store. So of course a lot of that tundra is in permafrost regions, so it's permanently frozen right now. And it's estimated that if you think back to the estimates from the atmosphere, something in the order of 1,672 petagrams, we probably can't be that actually precise, is locked up in northern permafrost regions in tundra. And again, thinking about vegetation and its importance in these regions, boreal forests are also important in terms of carbon, and one of the reasons that they're important is that they sequester a fair amount of carbon annually. So the estimates currently are that they sequester 1.3 petagrams per year annually. So the rates at which they uptake is important for understanding how much we can mitigate against fossil fuel emissions of carbon dioxide. So let's just have a think about what this means in terms of the Arctic. So this is a map of Arctic biogeographic zones, I suppose biomes, where there are different types of vegetation, and I'll be referring to some of these different kind of vegetation categories in this lecture. You can see that there's this enormous swathe here of kind of mountain tundra and northern taiga. So tundra ecosystems, I've just shown you sort of short sort of grassland to dwarf herb type vegetation. Where we go towards taiga, this is a kind of local term that's used to describe coniferous forest or conifer forest, pine forests, usually used in the Siberian region. And then as we get towards the middle boreal, that's more kind of extensive pine forests. So these terms, boreal forest, taiga, are used to denote these vegetation classes. You'll see that quite a large proportion of the Arctic is classified as Arctic desert, so it's got very little vegetation on it, and that's towards the poles, where you've got very little sunlight that reaches, so you've got very short growing season essentially. so very little chance for vegetation to grow. So it's quite clear from these patterns that vegetation growth is determined by the climatic sort of drivers that drive vegetation. And of course, if the Arctic is warming, we would suppose that that might change the limits that vegetation can grow. So this is an example here which measures how Arctic temperature has changed between 2001 to 2004 minus 1979 to 2000. And what it's essentially showing is that the increase in temperatures in the Arctic has been much greater than in lower latitude regions proportionally. So we've got this big temperature increase going on. The patterns in terms of precipitation are less clear, but we certainly have this kind of warming going on. And that's projected to continue. So these are projections for Arctic surface air temperatures, which are projected between the year 2000 to 2100. And you can see that even the most conservative estimates suggest that there'll be an increase of around about 3 degrees Celsius during that time. So warming is going to happen. It is already happening. And we need to find some way of estimating what influence that will have on Arctic carbon stores, and that's partly the work that we were undertaking. So one quite clear influence on Arctic carbon stores is this phenomenon of Arctic greening, and this has been picked up by a lot of the remote sensing satellites and things like that, which have been, so there's been kind of air photos, field studies, and studies like that, and also satellite data, which have looked at vegetation growth and tracked that the Arctic is becoming greener. And it's becoming greener because there's more vegetation growing there, but it's also the nature of the vegetation is changing. So those kind of woody vegetation classes are shifting northwards and changing the nature of the Arctic because it's warmer. So this, of course, has got the potential to alter carbon stores, and there's a lot of research going on by terrestrial ecologists looking at the effect that this has on the land surface. So some of that research has shown this is a measure of annual net ecosystem production. So it's really working out how much I suppose plant material gets produced on the land surface. And therefore, what effect, so if this goes up, you would expect that if more plants are growing, they're taking up more carbon dioxide through photosynthesis. So what seems to be happening is, since the 1960s, you've got this trend where you've got a strengthening of the so-called Arctic carbon sink. So carbon's being taken up more because you've got more productive plants growing on the land. But it's flattened off in recent years. So since 1995 approximately, there's been this flattening off where you've got this plateau, which is thought to be caused by the fact that a lot of these permafrost regions are starting to defrost. And once you defrost those frozen soils, they start to decompose. So that's kind of the reciprocal process as carbon fixation and photosynthesis. You get decomposition and release of CO2. So we'll be talking about some of these principles in this lecture because these are the things that really drive whether ecosystems and global systems are carbon sources or sinks. So we need to understand how Arctic greening is going to play out in the future. And one really key feature of Arctic landscapes are lakes. The reason why I've been focusing on Arctic lakes is firstly because I really enjoy working on lakes, and I really enjoy working on lakes in the Arctic. But they are a really important feature because we've got all this land surface here. Fine, you know, there's stuff going on there. We've got tundra, which is frozen for quite a part of the year, and it's in a permafrost landscape. Now, these lakes will be ice covered for something like sort of 9 months of the year, something like that, depending on where they're located. But underneath the ice cover, you've got unfrozen water. So that actually provides a vessel by which there's the potential for lots of biogeochemical reactions to go on, for biological activity to go on in an otherwise frozen landscape. So the importance of these sites within the Arctic landscape really has to be considered if we're thinking about how this landscape functions overall as a carbon source or a carbon sink. So I'm going to be focusing a little bit more on lakes for the rest of the lecture. I'm going to just justify why I think Arctic lakes are important. So one kind of key statistic is most lakes in the world tend to be really small. So we've got a few really big, enormous lakes like Lake Baikal, the Great Lakes of North America, but there's not many of them. But there are lots and lots of very small water bodies. And if there are lots of very small water bodies, that provides the potential for lots of interactions and reactions within these little reactive, non-frozen pockets of water to go on. And then the other thing is the distribution of these water bodies, which tends to be very strongly focused in the north. Part of the reason that it's strongly focused in the north is there's more land surface in the north, so it makes sense. There's more places for the lakes to be. But then also, it's partly because you've got glaciated landscapes up north. So it was once covered by glaciers, and as you probably all know, you're all geographers, right? So you probably all know that glaciations form lakes often. So that's part of the reason. There's also another type of lake, which we'll look at in a second, that can form in these kind of landscapes. So you can see it's a really important landscape for lakes in the north. So I mentioned this other kind of small type of lake that can form, thermokarst lakes. There's been a lot of work done on these sorts of lakes, and I won't be talking about this sort of lake for the rest of the lecture, but it's worth mentioning that one of the big concerns in terms of the Arctic carbon cycle is what will happen in this kind of a landscape. So covering really, really large swathes of northern Siberia, for example, that northern part of Russia, is landscapes like this. And they form when you've got really flat landscapes, you've got really large coverage of peat, organic matter, so old soils that have accumulated there. And then because of freeze-thaw action, you get the kind of collapse of these little sort of roundish ponds, which are very shallow, and so you get this sort of crazy-looking landscape. It's quite interesting when you fly over these kind of landscapes. So one of the big concerns is that now things are getting warmer, this permafrost landscape is starting to defrost. When it defrosts, it releases organic matter into the water, and that decomposes. If you've got a lid of ice on this, so this is the surface of a lake here. If you've got a lid of ice covering a load of organic material in a shallow lake and it decomposes, you quickly run out of oxygen, you keep decomposing and you push it towards production of methane, which is, you probably all know, is a very potent greenhouse gas. So this is always one of my favourite slides for lectures, which is they've literally drilled a hole in this lake, in the lake ice, and put a light to it, and that's all the methane burning off coming from this lake. So very potent methane source, and there's a lot of research being conducted at the moment into how permafrost melt will feed back and be processed by these standing water bodies. Okay, so those are thermokarst lakes. I'm going to talk more generally about lakes that are probably deeper than that and in more sort of montane landscapes. And I'm going to talk a little bit more generally about what we understand of lakes as carbon processing water bodies, I suppose, or bodies. And this is a model that was described, it was devised in 2007 by Jonathan Cole and other colleagues. And what it essentially tries to do is to reevaluate our understanding of how lakes process carbon. So what he was suggesting was you've got 0.9 petagrams. So the original model suggested that carbon passes from the land through the lake and to the ocean. Nothing happens. This model was called an active pipe model because what it tried to do was to describe how lakes process carbon, so that they played an active role in carbon processing. And this actually led to a re-evaluation of what we understand in terms of how lakes processed carbon, because what we realised was there's actually quite a lot more carbon coming from the land than we originally realised, 1.9 petagrams. Some of that was getting buried in the lake sediments, so sedimented out, but quite a lot of it was getting evaded, was being effluxed off as carbon dioxide. So what we were ending up measuring coming out to the ocean was only a fraction of what originally came from the land. So understanding how this bit works is quite important for understanding our sinks and our sources and how lakes behave in the Arctic landscape. Okay, this model's been revised actually, and the numbers that have been revised since suggest that lakes are more important than we originally thought. So they bury more carbon and emit more carbon than we originally thought. And so this suggests that lakes are probably more important than we originally gave them credit for. So this was the paper that suggested that. It suggests that the emission rates coming from lakes are similar to those in oceans, that the burial rates in lake sediments exceed those in ocean sediments, so they're very important for carbon burial, and that methane sources from things like reservoir impoundments and Arctic lakes are really important. So this paper was quite important in moving the science on and certainly helping us as researchers think, okay, we need to look at Arctic lakes. This is probably an important research area. Okay, so I've mentioned methane quite a bit. Most of the bit of the carbon cycle I'm going to talk about from now on really concerns carbon dioxide, and I'll be thinking about the processes really that drive the flux of CO2 from and into lakes, rather than thinking about carbon burial for this talk. Okay, so we've established this idea of Arctic lakes as kind of bioreactors really, so that you've got water in there that's ice covered for part of the year, but there's still stuff going on in it, and ice free for part of the year, and that can be a bioreactive vessel in which carbon related reactions can go on. And here are the key players in those reactions. So we've got algae, who are the photosynthesizers. So as you all know, photosynthesis involves taking up carbon dioxide, and therefore, if you've got more photosynthesis, you should increase your strength as a carbon sink, right? So you're taking up carbon dioxide. So if you've got more algae, more carbon dioxide is taken up. And then the reciprocal organisms. So these are microscopic organisms. These are in the plankton. See, this is a 20 micron bar. I don't have a scale on the next one actually, but these critters are much smaller. So this is a little nanoflagellate. But all these little dots are bacteria, and these are responsible for decomposition of organic material. So with photosynthesis, you build up organic material, take up CO2. With decomposition, you decompose that organic material and release CO2 by respiration. So if we have more decomposition, we'll have more carbon source. We'll strengthen our carbon source. So if we can look at the balance between those two processes, you don't have to look at the annoying sort of equation. The bit at the top, though, is what you really need to think about. We can think about net ecosystem production. So production is what's produced by respiration, by photosynthesis, sorry, and then you've got respiration losses to come from that, and that's net ecosystem production. And you can get respiration by your autotrophs and your heterotrophs, and we'll talk about what they are in a minute. If you have a system where you've got more respiration than photosynthesis, you will tend to, less respiration than photosynthesis. You'll tend to have emission of oxygen that's produced by photosynthesis and take up of CO2. If you've got more respiration than photosynthesis, you'll tend to have a carbon source, emissions of CO2. So this is just the sort of general rules of respiration and photosynthesis. And you may, so those of you who've done biology are probably familiar with the terms kind of autotrophic and heterotrophic. So autotrophs build organic matter through photosynthesis. Heterotrophs break it down. We sometimes use this term, net autotrophic versus heterotrophic. Now you'll notice that there's something going on here. There's an input which is governed by the amount of vegetation around these lakes, which is absolutely key in understanding Arctic carbon cycling, or carbon cycling anywhere actually in water bodies. So dissolved organic matter. Sometimes when you walk by a river or a lake, certainly in kind of peaty conditions where there's a lot of organic matter around, you might see that the water's stained very brown. And the reason it's stained brown is because material from the plants it's kind of breaking down. So this is dead material from plants and vegetation that is decomposing, and it breaks down into these things which are soluble but still organic. And therefore they're known as dissolved organic matter. So there's lots of different types of compounds that can be produced, or they can sometimes there is dissolved organic carbon, so DOM, DOC, right? Key thing is they're brown. This is really important for lakes. And the other key thing is they're a carbon source. So if you're a bacteria, you would really enjoy eating one of these molecules because it's lots of energy in it for you. Okay, the other main point about DOM is it's dissolved, right? So if you've got a load of vegetation in your lake catchment, it's not going to end up in your lake unless it's mobile. But if the vegetation has started to die, you know, at the end of the season, started to die, started to decompose and dissolves into the water, it can then get transported into the water. So, how does this relate to our Arctic situation? Well, if we've got more plants, we should end up with more brown stuff, more DOC. So that should have an impact on our lake ecosystem functioning. And the other thing that's probably going to happen as well is if we've got warmer conditions, we're going to change the hydrology. So we change the delivery of DOC. So we should enhance how much of it can go into our lakes. So let's just see how this plays out. I've been through most of these concepts, but these are our kind of models for suggesting what happens. So if you've got a nice clear lake with no DOC, this is what you'd expect to happen. You get lots of your photosynthesizers, your algae, and they're able to absorb the energy from sunlight, and that takes up CO2. You'd expect a strengthening of the carbon sink. You'd also, some of that will be deposited in the sediments. There's also, they need nutrients as well to fuel them. That's a side issue. That's something else that requires work. But focus on the DOC for now. Right, if we've got DOC, or dissolved organic matter, going into our lakes and turning them brown, what's the first thing we've done? We've turned the light off, so there's no energy from sunlight to drive photosynthesis. So your photosynthesizers immediately are not able to be as productive, and you've weakened your carbon sink. Well, the other thing you've done is to provide a carbon source which fuels your decomposers, so they like to break it down by respiration, and then it will lead to emission of CO2. So in this situation, lots of DOC, you would expect to have enhanced the carbon source from the lakes. And where does the DOC come from? Well, it comes from increasing the amount of vegetation in your lake catchment. So this is our model for trying to work out what's going on in lake ecosystems. Of course, it's not that simple in real life because you've got a little bit of sunlight getting in, and you've probably got a little bit of DOM, you've probably got both things going on. Photosynthesis, respiration, and what actually goes on in many lakes is your photosynthesizers then die, they get broken down by your bacteria, and then they release nutrients. And this is known as the microbial loop in lakes. So we're trying to unpick the relative balance which determines our lake metabolism and determines the strength of the carbon source or the sink. One other little group of organisms that are useful in indicating this are this group. And these are organisms that can either photosynthesize or decompose organic material. And these are known as mixotrophs. So not autotrophs, not heterotrophs, they're mixotrophs. So they can give an indication of how that ecosystem is functioning. So this is setting up kind of what's going on in Arctic lakes. So this is what we want to get at. We want to work out really what these CO2 fluxes are at the end of the day. Now there is a complication. So I've already mentioned that if you've got lots of brown stuff in your lake, you'll cut out the light. But the other thing you do is to change the quality of the light that reaches into the lake. So you might be familiar with this. This is simply a breakdown of the wavelengths that photosynthetically active radiation is in. So it's from 250 nanometers to 2,500. So you're right in the infrared range here. This bit is really damaging potentially to organisms. So the UV light can potentially play havoc with organisms. We know that. It gives us skin cancer, for example. So we need to protect ourselves from ultraviolet radiation. And one important photoprotectant in lakes is dissolved organic matter. So if we've got a lake, this is a lake up in Scotland, by the way, so this is me getting pulled around on a boat, which is the way you should be. I think, is that you on there as well, Nick? so you can see the water's stained brown, and those brown molecules of the dissolved organic matter act as a sunscreen. So it means that you can't get a whole lot of ultraviolet radiation penetrating too far into that lake basin. So the conditions within this water body are quite hospitable for the organisms in there. They're protected from UV damage. But if you've got this kind of situation, you can see there's not much vegetation in this catchment. This is a lake from a high elevation lake in Canada. Very, very blue water, hardly any DOC in it, and they're extremely transparent to UV. So UV damage is a real problem in some of these high elevation alpine lakes where you've got not much vegetation. So there are ways that algae can deal with this, that the organisms can deal with this. Here's an example from, this is a Himalayan lake where someone looked at these this is like a lawn of algae on here. And what these algae are doing, they're coloured that way because they're producing sunscreen pigments. So they produce their own pigments that protect from UV radiation damage. And that can tell us, if we can detect these pigments, it can tell us something about what the UV radiation, the transparency of the water is. Oh no, that doesn't look good. Restart required. see how we get on. So, okay, so here's our hypothesis that we want to test with the research that we've been doing. So I'm going to present some results from some of it's quite new research. We're just in the process of getting it published at the moment. It's just in review. And so what we want to have a look at is if we've got Arctic greening, so increased vegetation biomass, what is the effect on autotrophy? So how many photosynthetic organisms do we get growing, and therefore how well do we increase our carbon sink? We hypothesise a very low vegetation cover because you don't get much DOC, much of the brown stuff. You get UV damage, and that limits production. But here, if we've got really, really brown lakes, there's going to be no light to drive photosynthesis. So we think we're going to get this kind of unimodal distribution, and that's our working hypothesis for understanding what happens in lakes in response to Arctic greening. Now, the problem with understanding Arctic greening is it happens over really long timescales. So some of the best data sets that we have from the Arctic, for example, the place where I go work in Greenland, there's been a group that have been going there since I think the late 1980s, and they've been going every year to measure quadrats of the tundra vegetation, and they've got some fantastic data sets. But at the end of the day, it's only several decades worth of monitoring in one place. So how do you get around this when you want to understand processes that occur on quite long timescales? Well, we've had warmer periods in the past, and we can use these to tell us about what happens when we've got more woody vegetation. So this is an example of some work that was from is a collation of work from northern Eurasia, where they've essentially looked at remains from these different plants. Does anybody know the Latin names of these plants? Can anyone spot what these actually are? So we've got birch here, larch, and this one's spruce. So what this is telling us is that at certain periods in the past, during a warm period, the Holocene thermal maximum, we have this expansion in woody vegetation and then a subsequent decline. So we've got analogous periods in the past that we can use to tell us about what might happen if the vegetation gets, if vegetation grows more. So we can use this kind of approach as an analogue. We can use the past as an analogue for future change and to guide us in terms of what happens. So how do we reconstruct the past? Well, a lot of the work that I do and that also goes on within the School of Geography as well involves using lake sediments as archives of change. So you can see this is a nice example here because this is a lake from Iceland, which gives a really nice example of how lake catchments integrate all the material that from the catchment ends up getting washed down into the lake basin. Well, not all of the material, but a lot of it gets trapped in the lake basin. And then the main principle is that mixes together with the stuff that's growing in the lake basin as well. So we've got external, internal stuff. And then it sinks and is incorporated into the sediments. And because it's in time-ordered sequence, we've got a kind of archive that we could read back in terms of change. And if we take a core of that, we can then have a look at how this goes back in time. So this is an example of a sediment core that I sampled in West Greenland. I sampled this core in 1999, and I was immensely excited because it had beautiful laminations in it, and it's been one of my favourite sediment sequences. And I have to say, most of the cores that I've ever taken ever since have never been quite as good, so I probably peaked too soon with that. But so yeah, this is just an example of what very, very nice laminated sediments look like. But we have to decode that information. Okay, so we've got a sediment core, and we need to decode it. So the first thing we need to do is get our sediment core sampled. Now, I brought along a bit of a practical demonstration of how we do sediment coring. So in an Arctic lake, what you need to do is, first of all, it's much easier to do coring from the lake ice because you just walk onto it, but you have to drill through it. So these are two of our helpers in Greenland who have just cored through the lake ice using this ice borer, and they've just created a little hole here so we can get our coring equipment through it. And I brought this corer here. This is a brand new corer. We've just bent the old one, so we've just bought a new one. Right, so this is an example of one type of corer. There's lots of different types of corers. And this, right, I'm getting the right way around. So this is a so-called Russian corer or peat corer, but you can use it for lake sediments. And the general principle is, you have it like this, it's got a blade, and you lower it down, so it requires a little bit of kind of, you don't want to drop it, basically. So usually we have something attached to it to make sure we don't drop it. And then you push it into the lake sediment, and then if you do a quarter turn and it's actually in the lake sediment, so what happens is this blade traps the sediment within it, and you get a little semicircular amount of sediment which you can then pull up. And as you're pulling it up, of course, a lot of these lakes are 15 metres deep, so you've got all these rods that screw onto each other. So as you're pulling it up, there's icy water going on your head, and you're trying to get it unscrewed. And so Matt knows this; he helped with this when we were out in Greenland. And then you pull it up, and then with any luck, you go like that, and you reveal your beautiful sediment core. So that was the core that I just showed you was sampled in that way, actually. Okay, so yeah, lake sediment coring. So this is an example of what was going on in the field there. So this is we've got a large diameter Russian corer for this. So there's our sediment cores, and then this is what it looks like in a little bit more fine scale. So these are some of the cores that were sampled actually for this project, I think. Okay, now if we're going to decipher our sediment record, we really need to know where we are in terms of time. So one way we can date lake sediments is to use radiocarbon dating. Now, you've probably heard of this. It's a very widely applied technique. It's used a lot in archaeological studies, for example. And there's a big facility that we often send our samples to up in East Kilbride. where we can analyse radiocarbon using an accelerated mass spectrometer. So that will give us an estimate of the age of the sediment. So we just pick out samples here and get an idea of where we are in terms of age. Then if we want to know what was happening in terms of past vegetation, there's a couple of ways we can do this. Firstly, we can look at little remains in the sediment. So sometimes you get actual leaves preserved in sediment that have come from the catchment, or of course pine needles are quite resistant to decomposition, so they can just wash in, and that can tell you what vegetation was in the catchment. In situations where they're not so well preserved, often you can use things like pollen grains. So pollen's produced by male flowers, and it's quite broadly distributed, and you can identify them usually to the sort of genus level, and it can tell you what kinds of vegetation was there in the past. So we've decoded our lake archives to tell us what was happening in terms of vegetation biomass using plant macrofossils. That can help us to get at the first part of our hypothesis. But we still need to work out how we're going to work out what's going on in terms of autotrophy. So how much photosynthesis or carbon uptake is going on. So coming back to our diagram again, what we really want to get at, if we want to know about autotrophy, is these guys, the photosynthesizers. So we've got a way of looking for biomarkers of the photosynthesizers and the ones that are mixotrophic as well, the autotrophs and the mixotrophs. And so the biomarkers that we use is actually a real specialism that we have in the school, which there are not so many labs in the world that do this sort of analysis, which is we use chlorophyll and carotenoid pigments to tell us about what was there in the past in terms of these autotrophic communities. So the first thing we can look at is chlorophyll, for example. So you probably all know chlorophyll's green. And so that's part of the reason why most algae look green. And it's used for harvesting light in photosynthesis. But there are other pigments that algae produce. So algae also produce carotenoids, and they generally tend to be between yellow to orange to red in colour. That's partly what's causing the reddish colour in these algae. And their main role is to protect from light damage, and also to kind of feed energy into the main sort of chlorophyll pigments, to drive the photosystem for photosynthesis. Third interesting type of pigment, which I've already mentioned, is those sunscreen pigments. So that's quite useful for us to know about. If there's a lot of sunscreen being produced, that kind of suggests that the lake was probably susceptible to quite a lot of UV damage. So this is an example of those cytonemine pigments. These pigments tend to be reddish or black coloured, and what you've actually seen here is a combination between chlorophyll, which is green, and the blackish coloured pigments in those sunscreen pigments. You've got this quite dark green colour. Okay, so we've got a bunch of mud. I'm going to try a little experiment here, because I've got some mud here, and I'm just going to hope there's a little bit of pigment in the mud I've got. So this mud is actually from Malham Tarn. Has anyone been to Malham Tarn? It's classic geography site. In fact, yeah, we take our students there every year. So, so this is a sediment core, mud from a sediment core from Malham Tarn. And what I'm putting in here, I hope this kind of works, because I don't know. So I'm just going to sort of shake that up. So the idea here is I'm putting in some organic solvent. So most of the compounds that I just showed you are lipids. So they won't dissolve in water, but they will dissolve in organic solvents. So what I've got here is acetone, which is similar to what they use in nail varnish remover. So you can just put that into your pigment extracts, and this is the kind of colour that you might expect to get from your sediment core. Now again, this is from the best core I ever took example, and you can see there are these beautiful purple colours, orange, yellow. Again, I've never surpassed the beauty of the colours in that core, so we'll probably have a little bit more boring colour from Malham Tarn. I think we'll revisit it in a minute. So the idea though is that the colours in those extracts reflect a big mix of pigments. So you've got chlorophylls, different types of carotenoids, which are different colours themselves. So you can see you've got this blend of different colours, but what you need to do is to extract individual pigments or separate individual pigments in order to identify them. If you can identify them, you can tell what organism they came from. So this is the idea of biomarkers. They're marking a particular organism. So the lab that we have just up the way there has a high-performance liquid chromatography unit which extracts pigments and quantifies them and helps us to work out what we've got going on down our sediment core. Right, so now we can work out what was going on in terms of vegetation biomass. And we have a measure of autotrophy or photosynthesis using our photosynthetic pigments, which is directly kind of related to photosynthetic capacity. And this can allow us to work out relative net lake metabolism, net ecosystem production, and get at this idea of whether these lakes were carbon sources or sinks. The project that we were working on then was trying to test this idea over a range of different vegetation kind of densities. And so because we wanted to look right from tundra up to forest to look at different scenarios of vegetation change, we had to go to different parts of the Arctic. So we had a team in Southampton working in Alaska, we had a team based here and in Loughborough working in Greenland, and a team at University College London looking in Norway. And these covered different gradations across that kind of the vegetation transition. So we covered right from kind of herb coverage, which is very sparse coverage, right up to sort of conifer forest. So we generated a lot of these kind of diagrams, and don't worry, I'm not going to take you through lots of these kind of diagrams because I know they can be a little bit off-putting, but the general idea is we took our pigments, our carotenoids and our chlorophylls, which told us about the autotrophic production. We looked at pollen here, which told us what the vegetation was doing, and this is just summarising vegetation change. And then we did that at 6 sites from all around the Arctic, and then we wanted to see what the overall patterns were. So what we actually did was we took the data sets here and we standardised them for each core so that they were directly comparable. We put all of the data sets together to look at what the overall patterns were. So here's a summary of one of the really interesting findings that we found out at the beginning. So what I've got here is a gradation from very sparsely vegetated catchments up to very forested catchments, pine forest. And this kind of represents a dissolved organic carbon gradient, which means that the lake water is getting browner as you go that way. And the reason we know that the lake water was getting browner is because we've extracted these sunscreen pigments, the UV absorbing pigments, and we've scaled them relative to the amount of other algae, and this tells us that the algae, when the catchments were very sparsely vegetated, were producing more sunscreen. So they were exposed to UV, the water must have been really transparent, and therefore we've got some sort of evidence that there was possibly some UV damage going on there, whereas we don't get any up in this higher end of this scale. So what did that mean in terms of production? Well, we've got a pigment here from one of the groups of algae, the diatoms, and what we can see is, generally speaking, there was much lower autotrophic production, photosynthesis, going on by the diatoms when we have very sparse vegetation, which suggests that that UV damage was having a detrimental effect on the algae. Whereas once we've got a little bit of bit of colour in the water, it was protecting the algae, and this had a beneficial effect for production. And then as we go and the water gets darker and darker, this inhibits the ability for photosynthesizers to grow because you've got essentially really dark water, there's no light in it. And this was borne up a little bit because we've also got another pigment that indicates a mixotrophic species. If you remember, these are ones that can photosynthesize but also then utilise algae, break down organic carbon, so use heterotrophic carbon sources. And these increase, when you've got more of a carbon source, these increased. So that suggested to us that this was kind of being driven really by these patterns in in lake brownness, or the availability of organic matter in the lake. So what does this mean in terms of our conceptual model? So what we wanted to do was, we were testing a hypothesis, and then what we want to do from that hypothesis is to develop a model which can help us to understand what's going to happen in response to Arctic greening. So here's our overall predictions, and it does seem that at the lower end of the vegetation we've got suppression by UV. Here we've got light limitation as the water gets darker. But one thing we're trying to do is to make this more relevant. Okay, so at the moment I'm just talking about there's an increase or there's a decrease. So it's really hard to quantify, for example, carbon dioxide emissions using lake sediments. It's not possible. So what we need to do is move the science on and make suggestions for how we can get more quantitative about it. So what we know is under certain types of vegetation scenarios, we've got certain concentrations of brown stuff, RDOC, and so that means that if we look at lakes that are around about this concentration of DOC or this concentration, these are the points at which there are critical switches between autotrophy and heterotrophy. So that's where people who go and measure carbon dioxide fluxes from these lakes need to be going and measuring. So the other thing that we found is you've got this increase in mixotrophy as you go along this gradient, which makes sense. You've got more breakdown of this organic material. And then, so yeah, we've got essentially an increasing carbon sink at this end. So think about what this means in terms of Arctic areas right in the north where you've got very little vegetation at the moment. If they increase in terms of vegetation, this could have a beneficial effect in terms of creating a carbon sink. But further down south, where you've got forest coming in, you're probably going to create an increasing carbon source. The other part of this project, and I'm not going to show you this today, but what we're working on right now is then working out what this means in terms of carbon burial. So with our sediment cores, we can look at burial rates and work out how these patterns, what consequences they have for burial. So there's CO2 emissions from the lake and also burial, so that we can get the two kind of parts of that. Okay, so what I just want to finish off with is the work that we're doing now. So that project is just about finished off, the work that I've just shown you, and we're just trying to get it published at the moment. But I've just got a couple of slides of the ongoing research that we're doing right now. And what we're trying to get at is how terrestrial ecosystems are linked with lakes, but also the role that the ice sheet plays in Greenland. Okay, so you probably all know that Greenland has an immense ice sheet over most of its land surface. So this is a really dominant feature. And so one of the sort of a real sort of geomorphic feature when you go to West Greenland, so this is an area we've worked in for a long time in Kangerlussuaq, is this enormous outwash plain. So what this essentially is it's a river that receives meltwaters from the glacier that comes out just that way along from the ice sheet, probably about, I don't know, 30 kilometres, something like that. So obviously this is a seasonal thing, so this photo was taken, I think, early spring, so just as the melt was starting. And you can see what happens is when glaciers discharge water, the water is absolutely filthy. So glaciers have a lot of ground up silt and materials in them, which spill out on these rivers and get deposited as fine materials all along this outwash plain. When things dry out later in the summer, so this was a photo taken when we were out there in June. What you can see is, this is what happens. Get a windy day, all of that stuff gets resuspended. And so this is right down in that glacial outwash plain. And this was taken by some of our US colleagues who were out there at the same time. And we were just up the valley sampling this lake. So actually we could look down on this. You can't really see it from this photo, but we were standing here looking down at the dust storm that they were looking at down the way. And we were basically thinking, well, what's all this dust, what effect is it having on these lakes? Because the main point about Arctic lakes is they're extremely nutrient impoverished. So all of this photosynthesis and stuff that I've been talking about that goes on in Arctic lakes is still limited by nutrient availability. But if you've got dust landing on them that is potentially rich in nutrients, that has the potential to upset or change the carbon balance. So it's a very, I suppose, something that you might not think is inherently, an obvious driver, but it's something that could be quite important. So the project that we're doing right now is essentially trying to look at how much dust is being deposited and when's it being deposited. So we're doing some monitoring. We've actually set up some instruments which we're having difficulty getting working at the moment. So they're still out there. We're trying to get them working. But we've got a whole bunch of these things which are imaginatively called frisbee traps. They just essentially you collect dust. So we're going out and collecting those 3 times a year, and we've got this instrumented monitoring. We're digging snow pits to look at what settles on the lake and then gets melted out in the spring. And then this is actually just from about 1 litre of snow. Look how filthy it is. So there's a lot of dust being kind of resuspended and deposited on these lakes. We then want to know what's in the dust, what's it made of, what's the chemical composition. So we're running some leaching experiments that are out there right now doing their thing. And we've been sampling, this was me weighing out the aliquots for the leaching experiment, which is an extremely dusty job. I hadn't quite worked out that I needed a dust mask until I got in the lab. So we've been doing some sampling around there. And then we're trying to work out what impact it has on the lakes. So these are sediment traps where we've been looking at where the dust deposits in the lakes. These are enzyme assays. So we think that if you've got dust particles, that the nutrients that are in them probably need to be accessed by microbial breakdown. So we're measuring microbial activity to work out how much breakdown you've got. We're taking sediment cores to assess how much dust there's been in the past. And this is an interesting algae. This is a macroalgae called nostoc, which is a nitrogen-fixing species. So one of our hypotheses is that dust has a lot of phosphorus in it. So if you fertilise with phosphorus, you end up with nitrogen-fixing species because you end up with an NP imbalance. So we think that's diagnostic. Okay, so I'm done now. These are my summaries. So I hope I've convinced you that I think lakes are a very important component in Arctic landscapes, and it's really important to understand terrestrial feedbacks, but lakes really need to be included in that because they are such, they're these non-frozen components of Arctic landscapes. And then there is this potential for ecological surprises, such as these links between ice sheets, dust transport, and what have you. So this is the reason that we're continuing to do research in this area to try to quantify that. So I'll just finish off by, there's loads and loads of people helped us over the years with this work, so those are some of the people I want to credit with that. Okay, thank you. There we go. Melon tarn. So it's kind of, greeny orangey. Some chlorophyll in there. You can come and have a look at it later if you want.
I really want to thank Suzanne for a really enthusiastic lecture and some great information there as well. Now's the time to see if you've got any questions though. Quite a lot of information there. Questions for Suzanne. It's a hard one sometimes.
Yeah.
Oh, yes, we've got one at the back, Marnie.
When are you next going back to Iceland?
Greenland. So we're going in April. So we go once when the lakes are covered with ice, and then twice when the ice has come off. So we go mostly in the growth season. Otherwise, it's a fairly bleak winter trip. So we'll do snow sampling to look for the dust on the snow in April. Yeah.
I'm sure Suzanne will be around for
Yeah.
maybe.
Come and look at the pigs then.
Come and have a look at the pigs. and that rather dangerous symptom that you've got there as well. Not many